Okay, so welcome to this next video on the G2M transition. At the moment we're just making our way through the cell cycle so that we can add the G2M transition onto what we already know, basically. Okay, we have discussed how the E2F transcription factors, along with their dimerization partners, are going to increase the expression of cyclin A, which is going to mean that we get cyclin A CDK2 complexes forming, basically. Okay, so we now need to look at what is the function of these cyclin A CDK2 complexes, which are also known as this S-CDK enzyme. Okay, so basically they are going to alter these complexes of proteins that we've formed at the origin of replication. So let me just remind you of what we've formed so far. So let's say this is an origin of replication here. Okay, and we've seen that so far we have the protein origin recognition complex, or ORC, right at the center of the origin of replication. Then we have these four other proteins which are also part of this great complex, two of which are CDC6 and two of which are CDT1. And then to the side we also have these MCM helicase proteins here. Okay, so this was what we called the pre-replication complex. So let me just color everything in and label it. So in blue, we have this MCM helicase here. Okay, so this is the MCM helicase. MCM helicase. Then in the center, we'll color in the origin recognition complex, or this ORC protein, which has which was the first protein to bind, basically, when we formed this pre-replication complex, and it recognizes the origin. So this is an origin recognition complex. Okay, and then over here we have this CDC6 protein. So this is CDC6 here, and then we also have CDT1 on the off diagonal here. Okay, so CDT1, we have two CDT1s, and we called this the pre-replication complex, CDT1. There we go. So this was overall the pre-replication complex, and in the G1 phase of the cell cycle, we have produced these pre-replication complexes on all of the origins of replication on all of the chromosomes in our nucleus of our cell, basically. Okay, right. So now what has to happen is you have to alter these pre-replication complexes in order to get them ready to actually dock a DNA polymerase enzyme and actually begin the replication of the DNA. So the first alteration that occurs is not actually done by this S-CDK at all. Instead, it's done by an enzyme known as DDK, which is another kinase enzyme. So this is DDK. And basically, it's going to phosphorylate these MCM proteins. So the MCM helicases are going to get phosphorylated. Okay, so I'll draw that first, and then we'll see all the transformations that this S-CDK is going to make, and that's the important bit. Okay, so here is this first alteration to the helicase enzymes that has been made by our DDK enzyme. Okay, so it's added a phosphate group onto these MCM helicases. Apart from that, everything else is the same. So here's our origin recognition complex, here's our CDC6 proteins, and here's our CDT1 proteins. Now, let's discuss what the S-CDK, or this cyclin A CDK2 complex, does to these pre-replication complexes now. Well, basically, it does how many major things? Two major things, I think. Firstly, it's going to phosphorylate these CDC6 proteins, and when it phosphorylates those CDC6 proteins, they fall off the pre-replication complex. So the CDC6 proteins are going to come off. So they're going to be phosphorylated, and they're going to cleave off. Okay, so they're going to come off over here. So let's say these are our CDC6 proteins, which have now got these phosphate groups attached to them. Okay, when the CDC6 proteins come off, then you're also going to get the CDT1 proteins coming off. So all four of these are going to dissociate, basically. So these are our CDT1 proteins coming off as well. Okay, and that's all the work of S-CDK. Now, it does more. It hasn't finished yet. S-CDK is also going to phosphorylate two proteins known as SLED2, SLD2, and SLD3. Okay, and when it phosphorylates these two proteins, their activity is turned on, basically. So it's going to add phosphate groups onto both of these proteins. Okay, and what these two proteins are going to do is they are going to majorly alter the pre-replication complex. So we've already had the CDC6 and the CDT1 proteins fall off. Now what's going to happen is these SLED2 and SLED3, which have been activated by our S-CDK, are going to bring in a majorly important protein onto the pre-replication complex. And this majorly important protein is called the pre-initiation complex. So it's not a single protein, it's a whole complex of proteins. Okay, so this is the pre-initiation complex. Right, so what colour shall I draw that in? I'll give it in this red colour, this important red colour. Okay, so let me show you what's going to have happened to our pre-replication complex now. So the CDC6s, these things here, which I feel like I should colour in because otherwise everything's sort of falling apart. So these are the CDC6s. They are going to cleave off, basically, and following them is going to come off these CDT1 proteins as well. Now, the origin recognition complex in the centre and the MCM helicases, which have been phosphorylated by this DDK enzyme, are going to remain. But you're also going to add in this massive great pre-initiation complex, which is going to start causing the DNA to open, basically. So what's going to happen is your origin of replication complex is going to open like this. So the two DNA strands are being pulled apart, and these great big pre-initiation complexes are involved in this. They're holding them apart, basically. Okay, and you bring in two of these. I should have stressed that. You bring in one on each side. So here's one, and the other one is over here. Right, now you still have these MCM helicases, but they've gone their separate ways. So they used to be attached to both strands. Now they're only attached to one. So here's our two MCM helicase enzymes. Okay, right, so let me colour those in. So here are our MCM helicase enzymes, here, and I'll colour them in here as well. Right, so they're now attached to different strands, and you've got these huge, great pre-initiation complexes bound and holding your DNA apart, basically, like so. Okay, right. The other important thing is that you recruit another origin recognition complex. So this origin of recognition complex can only now bind to one strand. So what has to happen is you bring in a second one, basically. So you now have an origin recognition complex recognized on each of these two strands. Okay? So this is what overall has now happened. You've altered the pre-replication complex so that they now form this. And what can now happen is that DNA polymerase can come in here and begin the process of of replication, basically. So what will happen is that DNA polymerase will come in. So let's say this is DNA polymerase. It will come in and it will basically begin the process of copying the DNA. And what you'll end up with, basically, right at the end, is you'll end up with two identical DNA strands. Let's say these are the two origins of replication that you've now copied, and what will still be there is the two origin recognition complexes. So one on here and one from here. Okay, so you'll copy the DNA, and that's the process of synthesis. Right, so let me just colour code these DNA strands so that everything is completely clear. So what colour should I use? Let's use pink. We haven't used pink. So this pink strand here, this is this strand here, okay? So that's that origin recognition complex from that pink strand up there, okay? And then we'll use orange as well. So this orange strand below here this is this strand here. So these two new strands have been synthesized by the DNA polymerase enzyme, basically. And it went, I want to stress that it went in one direction from this origin of replication. It did not go in both directions. You did not send one off in this way and the other off in this way. It went both this way. It went along both strands and synthesized these complementary strands coming off from here. The origin of replication on the other side will have come along here and synthesized these other two bits here, basically. Okay, and when you actually set off your DNA polymerase and it starts copying the DNA, there's a bit of vernacular that you should know about that. That's called the pre-replication complex firing, or the origin firing. So this is said to be the origin of replication firing, origin firing. Okay, right, and that causes the copying of the DNA, the replication of the DNA. Right, so I just want to finally discuss what happens to these two proteins here, this CDC6 and the CDT1 protein. So basically, you don't just phosphorylate CDC6 by S-CDK and it just goes off and just no longer associates with the pre-replication complex. No, because you need a safety, you need some sort of safety measure. You see, there's a great problem that could happen, basically, which is that once you've made all the proteins associated with DNA replication, what's to stop them all coming and associating again down here, i.e. in the same lap of the cell cycle? What's to stop all of these proteins coming and associating again on these origins of replication here, and then setting up replication complexes again, and then firing and copying the DNA multiple times in the same cell cycle, which is not what you want. You want the DNA to copy and you then to split in two. You do not want the DNA to copy and then it to copy again and have this cell with 4 copies of the DNA in. So you need to destroy some of these proteins that are associated with building these pre-replication complexes. And the ones that go are the CDC6 and the CDT1. So, CDC6 gets it worse than CDT1. CDC6, once it's got this phosphate group on, basically is ubiquitinated. So it has a ubiquitin group added onto it. So this phosphate group basically targets it for ubiquitination. So this is a ubiquitin group. And basically, proteins which get ubiquitin groups on get destroyed by the proteasome. So the proteasome is quite a formidable thing in the world of cells, well, in the world of proteins. It's basically a tube. So let me show you a little picture of it. It's basically a tube, and proteins go in one end, and amino acids come out the other end. So this protein, the CDC6 protein with the phosphate group and the ubiquitin group added on, is going to go into the proteasome and it's going to be destroyed, basically. So that's how you ensure that you cannot replicate the DNA more than one time in a single lap of the cell cycle. In addition, something also happens to the CDT1 protein. It doesn't get destroyed like CDC6. Instead, it associates with another protein which inhibits it. And this other protein which inhibits it is a protein known as geminin. So this protein which is going to bind to CDT1, and inhibit it until the cell cycle, until you're on the next lap of the cell cycle, basically. This protein here, which I'll colour in blue, is known as geminin. Okay, right. So, that basically is how the S-CDK moves you from, takes you into the synthesis phase, how it triggers the replication of the DNA. And we've seen how we go from the G1 phase into the G1S phase, and then into the S phase, and then to copy the DNA. Okay, so now we're ready to see, we're ready to talk about the next phases of the cell cycle, i.e. the G2 phase, and then the transition from G2 to M phase.